To inoculate a Petri dish, in order to isolate a bacterial colony so that you can perform your gram stain, we're going to be doing what's called the streak technique. First, you get your Petri dish. This is TSA agar with 5% sheep's blood. And with your Petri dish on the back side, I've already drawn my lines for my 3 zones. You can take a Sharpie and just make a mark similar to that on the back. Next, you're going to collect your patient sample. You want to use a sterile cotton-tipped applicator, such as a culturette. Be sure to maintain the sterility of this swab. Take this swab and use this to collect sample from the patient. If the area that you are swabbing is dry, be sure to moisten this before you collect your sample. Ideally, you would moisten it with sterile saline, but since we're doing this for class purposes, it's okay if you go ahead and moisten it with tap water. So we'll collect our sample from our patient. Now our swab is coated with our sample. So you want to take your Petri dish, and the first zone that we're going to inoculate is going to be this larger zone. So you take your swab and you want to roll it back and forth in kind of a serpentine or a zigzag motion until you've covered the entire zone of the Petri dish, which is about 1/3 to 1/2 of the Petri dish in total. It's important to roll the cotton tip applicator back and forth rather than scraping, because you can very easily damage the agar, which will crack and kind of shrivel up or shrink during incubation. So now that side has been inoculated, I'm going to go ahead and discard this swab. And next, I'm going to pick up a sterile inoculation loop. And what I'm going to do is lay it down, and I'm going to drag sample from the dirty zone over into the clean zone, just this top zone here. And I'm going to do this multiple times, just until I feel I've coated it completely. Now that I've done this, this loop is dirty. I'm going to discard this, and I'm going to get a second sterile inoculation loop. And I'm going to rotate my plate, and this loop is going to drag sample from this area to this third and final zone in the same fashion. Once that's complete, you want to replace the lid on the Petri dish. You can use a piece of Scotch tape to hold it closed if you so choose. We're going to flip it upside down. With a Sharpie or a marker, you want to write your name and the date, and also the sample source, so what animal it came from and what part of the animal it came from. And then we're going to place these in the incubator for a minimum of 48 hours. What's most important is to make sure that when you place these in the incubator, they are upside down, because condensation is going to form, and if it was right side up, that condensation would drip and it would pretty much flood the agar and no bacteria would be able to grow. So make sure to place it in the Petri dish upside down and wait for it to incubate. After it's been fully incubated, it's going to look something like this. So you can see the zones that have been drawn, and the larger zone, which was the one that we inoculated with the dirty Q-tip that came directly from the patient, you can see that had the most bacteria in it. And a lot of the colonies are overlapping and touching, and it would be really hard to isolate a single species of bacteria from that area. But this zone, this was the second zone that we had spread our sample into, and you can see that the bacteria are not quite as crowded, but still some are growing on top of one another. But the third and final zone, you can see we have some nice individually spaced out colonies, which will be easy for sampling when we do our gram stain lab. So when you've done your streak technique properly, this is pretty much what it should look like after you've incubated it.